When we talk about CRISPR system, we talk about guide RNA, we talk about CRISPR RNA, and we talk about Cas9. However, we tend to forget about the PAM sequence. PAM sequences are one of the most underrated sequence of the CRISPR system. Without PAM motif, the whole CRISPR system would fail, will not work. Welcome everyone to the new video in the series The CRISPR Craze. Today we'll be talking about PAM sequence or protospacer adjacent motif. Commonly we call it as PAM sequence or PAM motif. Let's talk about the structure of PAM sequence. PAM sequences are 3 to 6 base pair in length and they are located towards the 3' end of the guide RNA, which is called protospacer side. Therefore, they are commonly called as protospacer adjacent motif because they are located right next to the guide RNA in the 3' end. There is no single PAM sequence. PAM sequences are based on their nuclease. Each nuclease has their own PAM sequence. For example, let's take Cas9. Cas9 is derived from Streptococcus pyogenes bacteria. Don't confuse, it's not my name, CIRSPR SP. It's Streptococcus pyogenes. The PAM sequence for Cas9 is NGG, where N represents any nucleotide. So it could be AGG, TGG, CGG, or all three G, G, and G. These are the PAM sequence for Cas9 nuclease. Now let's talk about the application of PAM sequence. How PAM sequence are used in CRISPR system. So whenever there is a CRISPR system involved in an organism, the nuclease, it scans for a PAM sequence. Whenever it finds that specific PAM sequence, then it will look for guide RNA sequence. If there is a match, then there will be only a complementary base pairing between the guide RNA and the target DNA sequence. But for that, there should be a PAM sequence located next to the 3' end of the complementary guide RNA sequence. Otherwise, the Cas will not go to that location. Finally, when the guide RNA sequence and the PAM sequence match, the CRISPR will attach to that location and it will cut the DNA and the whole CRISPR system works. Thank you for watching this video and I'll see you in next video of the CRISPR Craze lecture series. Thank you so much.